2.
Hi, everyone. My name is Stephanie, and I, along with my teammates Reagan and Jeremy, performed an analysis of shared predatory toxic behavior within fungi of similar taxonomy. So the whole impetus for this project was in a paper that was recently published that showed Pleurotus ostreatus, or oyster mushroom, releases a compound known as 3-octanone that contributes to carnivorous behavior towards predatory nematodes. The ketone is produced from the lipooxygenase, or LOX enzyme, and so therefore we utilize sequences from LOX enzymes as kind of an approximate to judge shared predatory toxic behavior. And what we found is that LOX enzymes are very similar and found amongst highly dissimilar species. And P. ostreatus strains themselves actually showed greater LOX similarity for nucleotide sequences. And overall, the Pleurotisae family showed high similarity for the LOX enzyme and ultimately suggesting a possible shared carnivorous trait. And just a little more into introduction and background, Pleurotus ostreatus, the oyster mushroom, is found across Asia, North America, and Europe. And it is edible and renowned for some of its nutritional qualities as well as some bioremediation properties. And ultimately, this paper that was recently published showed that these mushrooms are able to have a carnivorous behavior on nematodes. And thus, our study aims to compare P. ostreatus to other similar taxonomic species to determine if that toxic behavior is shared using resources such as BLAST.
So for our methodology, the first thing we did was we identified potential LOX enzymes. You can look in our reference section for the list of enzymes that we found. And we did a blast of these to compare them to our fungus of interest to see if there was alignments that occurred. We then took those blast alignments and we ran them against the 5 strains of the fungus to our POLX enzyme in a nucleotide blast. And then we looked at the significant alignments for those. Then we did the exact same thing, but with a protein blast against the fungus strains. And the next thing we did was we examined the taxonomic tree for the fungus of interest. And we wanted to go up the tree through the species, genus, family, order, class, and phylum. And we blasted those against the POLX1, looking for Cori coverage to see if there was a similarity and if this was due to an evolutionary stance. And then finally, we did a T-blast N of the POLX enzyme against other species to see if there are similarities of Cori coverage and the relations that occur.
So the results from our experiments do tell us 3 things as they relate to the chemical 3-octanone that's as a result from the LOX enzyme. The first is that there are multiple LOX enzymes found in different organisms that are similar to our organism. They have similar mRNA, the similar mRNA POLX1 found in our fungus, as demonstrated in table 1. Second thing we found out is that the POLX1 enzyme did have more significant alignments when we did a nucleotide blast versus a protein blast of our organism and in similar strains. And the third thing we found was that other species within our organism's taxonomy bracket do have significant alignments as demonstrated in tables 4 and 5. So kind of the implications for this and kind of what we thought originally with our hypothesis is that other organisms within our organism's taxonomy bracket actually do have similar genes that do allow them to demonstrate a similar toxic behavior. And that is what we found from our study using BLAST-N and BLAST-P for our organism versus other organisms in its taxonomic bracket.